disease. When you are able to study the pathogenesis of that disease, you will get adequate information about that disease. So if you pick BP as a case study, you have to study the pathogenesis of high blood pressure. You look at the mechanism of that disease. You study in 3 ways. You can do a study of that disease in 3 ways. You can decide to study the molecular process, the biological process of that disease. So when you make a critical study on the biological process of that disease, the next thing you do is to study the molecular function of that disease. So you look at which enzymes play role, which enzymes, which genes play major role on that disease at the molecular level. Then you have to study the cellular component, which part of the body, which part of the body does that disease, which area in the body does that disease usually attack. So when you look at the biological process, we are studying the pathway of that disease. Then when you look at the molecular function, we are studying the genes, the enzymes that are associated with that disease and their role. Then when you study the cellular component of that disease, you have to look at, okay, which part of the body, which organ in the body does this disease usually attack? Does it attack the liver? Does it attack the kidney? Does it attack the pancreas? Is it at the testes, the ovaries? So you look at these things. These things are what you would do personal on your own. You do that personal on your own because it will guide you on the approach you're going to use for that particular disease. So when you have done this part, the next thing you look at is still from that biological process, you now look at the pathways, the possible pathways of that disease. So if we are looking at BP, you look at what are the pathways of BP, the high blood pressure. What are the pathways? Then in that pathways, which enzymes are involved? So that is where you now get your targets. And you know, the issue you may have is that there is no one target for a disease. Every disease has multiple targets, multiple pathways. So there is no one pathway for a disease. So you are going to choose which pathway do you want to target. Of course, you may not get a drug that will cover all the pathways, but you may just look at the pathways that are actively involved in that disease. So when you be able to get the pathways that are involved, they look at the genes that are replicated in that pathway. Those genes in that pathway is what will form your targets. Those genes now form your targets. So how can we achieve all this? For now, you can leverage on literature. You can leverage on literature because you look at some of the targets reported by other authors. Yes, you look at some of the targets reported by other authors who must have done work on diabetes, on high blood pressure. Then from there, you can see the targets reported in that work. So you can use it as a guide. You can use it as a guide for your own. Another way you can get this target is when you go to some bioinformatics database and type that your disease. When you type that disease, then you will see some of the receptor, some of the targets of that disease. You can now from there choose the one you want to work on, but you must identify the pathway you are targeting. Very important, because you must explain the mechanism of that pathway. But these two process is not advisable. I don't usually advise using literatures. It's still good to see what people have reported, and then using a database to search for your protein. The best approach to actually identify your receptor for that disease is through the use of network pharmacology, and that is a whole lot on its own, actually. So it's through the use of network pharmacology. In that network pharmacology, you'll be able to identify the genes that are actively involved in your disease. And what you do there in network pharmacology is that you are going to identify your compounds, the compounds you want to work with. So when you identify those compounds you want to work with, you have to also identify the genes that those compounds has the ability to inhibit or activate or modulate. So you have to predict the genes, the target, the possible target of those your compound. Now, after when you have predicted the possible target of those compound, you have to also predict the possible target of your disease. So this is the best way where we can get our receptor. Now, I have 5 compounds, and these 5 compounds, I want to use them against high blood pressure. So each of these compounds, I predicted their possible genes that they can attack. Maybe I got total of 600 genes that these compounds can attack. Then I'm working on high blood pressure. I have to also look out for the genes expressed during high blood pressure. So the next thing I will do, I will check these genes that my compound can attack. Are they also found in the high blood pressure? So I will now do a Venn diagram work that will check for common genes. So if I can get the common genes, it means that those common genes are the genes that I will focus on. Now, imagine if I use a gene that is not among these common genes. It simply means that those my bioactive compounds may not be compatible or may not attack those, may not interact those genes very well, because those genes are not compatible with my compound. The compound doesn't have business with those genes. So if you are searching by literature, you may end up searching those receptors that the compounds in your plant don't even have business with. And if you are going to database to search in database, to also search in database, you may end up achieving some proteins that your compound does not have business with. So that is the whole idea of that network pharmacology. It helps you to identify the genes or the receptors that your compounds can actively interact with. So it will just look at the common genes. So imagine that out of the 600 genes in your bioactive compounds, and maybe out of 5,000 genes from your disease, 10 genes from the bioactive compounds we are also found in the high blood pressure. So it means that if you can focus on these 10 genes, 10 common genes, then you are at least you have a better result, because the findings just show that my compound can target these genes, and these genes my compound can target are also found in high blood pressure. So your result will be awesome. But we will not look at network pharmacology in this training. Yeah, we may not look at it in this training because it's a whole big work on its own. If we bring in that network pharmacology in this training, we may not really be able to achieve it in 1 month. So we have a training on network pharmacology. It's still part of our monthly training. I may not know when that training will be on, but just be out for network pharmacology. Whenever you hear a call, you need it to seal up your work on pharmacognosy. So what we shall just do today is to leverage on literatures, see what people have done. Like I said, it's a probability that, yeah, you may end up using proteins that are than that your bioactive compounds doesn't really have much business with, because bioactive compounds doesn't have a good binding affinity with those receptor. But that is what we'll just do. We'll leverage on people's reports, and it's a 50-50. So we'll leverage on people's reports, get some literatures, see the protein that we are reported, and then use them. Now, because we are leveraging on literatures, I usually not advise when we are using literature reports, or if you are working with database, I usually advise that you don't work with one receptor. So even if you are doing molecular docking, you don't use one receptor. That ideology where you use one receptor is totally wrong. At least you use up to at least five receptor, and this five receptor must be from different pathways. Like I said, from different pathways, because there's what we call polypharmacology. Drug is has multiple targets, so we call it multi-target. So drugs has multiple targets. Even your disease too has multiple targets. So while choosing your protein, when you have been able to identify the pathways, you look out for for receptors on those pathways. So that's the first thing you will do, and I've explained how you can do that. So if you are leveraging on literature review, you have to identify any protein you identify. You study the pathway of that protein with respect to your disease. You study the pathway of that protein with respect to your disease, and that is number one. Then number two, you must have a control. So you must have a control, and the control, most times I see docking work where you see a docking study one control for different receptor. That is not good. That is not good. Now, the control drug we use, the reference drug we use as control, we also depend on the pathway you are working with. So, for instance, if I want to do a work on anti-inflammatory, and I have a protein, a cos one, maybe I'm using a cos one protein and a cos two protein. So these are different mechanisms. Now, since I'm using two protein, a cos one protein and a cos two protein, if you look at some of the work online, what people usually do is they will just use ibuprofen or aspirin or any of these NSAID. They will use only one drug as control for these two different enzymes, for these two different pathways, which is not ideal. So you have to look at for controls, drugs in the market that target the same pathway you are working with. So if you are working with 5 proteins and each protein represents one pathway, then look for drugs FDA approved drugs in the market that target each of those pathways. At least you must have two controls. You must have two controls for each of your receptor. You must have two controls. I would even appreciate having up to like 5 controls to make the work robust, and I think they made the work very good to look upon. So you can have like 5 proteins, or at least 2 proteins, sorry, 2 controls, 2 controls. So these drugs you are using as control, we call them positive control. They are positive controls. So if you are doing your docking now, you have identified the protein you are using. The next thing you do is to identify the control that you are going to use. And there must be a positive control. Now, that positive control is a reference drug that attacks that has been used for that same pathway you are working on. That will form your positive control. Then we also encourage that there must be a negative control. Yes, in your molecular docking studies, there must be a negative control. Now, that negative control is a drug that does not have business, a drug that does not have business with the disease you are working on. Maybe since you are working on high blood pressure, and we have a negative control, which can be a drug used for ulcer, or a drug used for a disease that does not have anything in common. Probably I can look for a drug for Alzheimer's disease. I can look for a drug for Alzheimer's disease. So that drug I'm using will serve as my negative control. The reason is for off-target interaction. So I want to know if my compounds, since I have a drug that is not made for BP, so I will compare the off-target interaction of that my compound with respect to the drug that is not meant for BP. So now, but there are 3 sets of control that must be in your docking work. One of the control I've explained is the positive control, which is from a reference drug. Then negative control, which is from a disease that does not have any relation with your study. That is for off-target interaction. The essence of that positive control, which is a reference drug, is for comparison. I want to compare this my formulation with standard drugs in the market, if mine can have a better binding affinity compared to some of these standard drugs in the market. So that is the essence of the positive control. Now, the third control that must be in your work also is your ligand, your native ligand, the native ligand control. Now, we don't use proteins that does not have native ligand. So that protein you want to use for your docking, you must check if the protein has a native ligand. So you must check if the protein has a native ligand, if the protein is co-crystallized. So if the protein is not co-crystallized, then you don't have business with such a protein. Except there are times when, like there's a work I did on that work was done last year. It was about worm, something on fluorescence. Now, when I was doing that work, I could not find a protein for that disease I'm working on. There was no protein. Of course, not all protein has been modeled in some of these bioinformatics database. like protein data bank. Not all protein has been modeled. So in the case whereby you encountered a problem that you have searched in literature, you could not see enough protein. You have searched in database, there was not enough protein. What do you do at that point? What you just have to do is to go through the genomic way, where you have to get samples of that disease, though some of them appears to be very difficult to get. To get samples of that disease, you do your molecular characterization of that disease, then you do your genomic study, get the amino acid sequence of that disease. So when you do the DNA extraction and others, so when you're able to get the amino acid sequence, we can now use what we call homology modeling. We also have a training for that on homology modeling. We can now use homology modeling to build the protein yourself. All the protein we see in protein data bank, we are built. They were modeled. So they were modeled. So we now model the protein from the amino acid, sequence. You can use Swiss model, you can use modeler, you can use AlphaFold 2 or AlphaFold 3. They are used for modeling, for protein modeling. We call it homology modeling. So when you now model the protein, now because you model this protein, there was no native ligand. But while you're modeling the protein, we usually have a template. That template is for comparison. And that template must be a protein that has great similarity with the one you're about to model. So you look out for an organism that is similar to what you're working on, then use the protein of that organism as a template. But that organism must have a native ligand. The one you're using as a template must have a native ligand. So you use the native ligand of that template depending on the similarity. But you use the native ligand of that template as the native ligand for your new protein that you have just modeled. Because not all of these proteins are found in protein data, but not all has been modeled. Now, even when you do network macrology, you get your common genes. Most times when you take those genes to protein data bank, you discover that their protein does not exist. Their protein does not exist. So you still have to come back to homology modeling to use the amino acid sequence of those genes, those packaged genes, to model the protein. So these are some of these things are what you encounter. So we still have a training coming up on homology modeling, which I may not know the exact month, but it's part of our calendar for this year. So you need these two courses now, network macrology and homology modeling in terms of your protein. So you must use a protein that is co-crystallized. The essence of using a protein that is co-crystallized is for validation, because the native ligand, the position of that native ligand in your protein defines the active site. Because the people that model that protein, they used that native ligand, that co-crystal ligand, to validate the protein. So wherever the native ligand is attached to is presumed to be the binding site of that protein. So that means every interaction that must be done on that protein is expected to bind around that binding site of the native ligand. So we use that native ligand for validation. We call it superimposition. So you shall now superimpose the interaction of your own ligand with the native ligand to see if they interact at the same binding site. So if they do, that means you have a better interaction. So these three controls must be there alongside with your bioactive compounds. So they must be there alongside with your bioactive compounds. They must be there in your docking. So far, we are going to now take hands-on on what we have discussed. But then, let us prepare our ligands. Do you have any question so far? So we can now do what we have discussed. Any question? Hello? Okay, I think no question, right? I don't know if you can hear me.
Yeah, somebody has raised
We
hand.
can hear you, sir. Hello, sir.
Hearing you.
We can hear you, sir. Hello, sir.
Okay, yeah, I can hear you.
Yes,
Yeah,
sir. Yes,
building.
sir. Good evening, sir. Good evening, sir.
Yeah,
Thank you, sir. Yes, sir.
yeah,
Yeah. Now, my question is based on the positive control, the reference drug.
yeah.
Can we also make use of our co-crystal ligand as a reference drug? Is it advisable?
Okay. Most times those pro-crystal ligands too are drugs. Yes, those pro-crystal ligands are they're also drugs. They are
Yes,
FDA
yes.
approved drugs. Yeah, some of them are FDA approved drugs. So, but I
Mm.
will advise you look for another reference drug still
All
on
right.
that mechanism and attach it so that let that so that you end up having a native ligand, a positive control, and a negative control.
All right,
Yes.
sir. Thank you, sir. Second question, sir.
All right.
Yes, sir. During there are some software like Schrodinger now. During preparation of the proteins, sometimes you don't normally remove the co-crystal ligand. Are they going to be removed automatically during docking process?
Yes. In as much as a master, the container you use does not require you to remove uh, the native
Co-crystal
ligand.
ligand. Yes,
Yeah.
sir.
Yeah. It means that it's its presence based on the algorithm built with a screenreader, it will not affect your interaction with your own ligand.
All
So in
right, all right,
fact,
all
the
right.
presence there is to define the active sites.
All right, all right, all right, sir.
Yes. But
But we used to
we're
remove it
using
for pellets.
Pyres. We're
Yes.
using Pyres. So because we're using Pyres, we
Yes,
are
sir.
going to prepare the ligand by removing the cold crystal ligand.
Oh, all right. Thank you very much, sir.
All right. So yeah, do we have another question so we can balance it up before we go to hands-on?
Yeah, before we go into answer, I have a clarification to make or a question. May I go ahead?
Yes, sir.
Yeah. I know that in silico has its own limitations. There is a particular scenario I want to bring on the platform and I see whether in silico will be able to handle it. There are certain receptors or enzymes that have multiple binding sites. There is a binding site for the ligand, and there's another binding site for what we call modulators. That's, these are modulators that can, change the conformation of the binding site of a receptor and begin to affect the affinity of that receptor to a ligand. The typical case that we've been talking about is like the receptor has just one binding site for a particular ligand. But those receptors that are or enzymes or proteins that have multiple binding sites, how are they handled during docking? I hope my question is clear.
Yes, it's clear. Uh, that is a wonderful question. there is no protein that has one binding site. Yes, there's no protein that they will say, okay, that this receptor, this is the binding site. It's just that from the protocol, from the protocol, we narrow down the binding site of that protein at the same binding site of your native ligand. Now, but that doesn't mean that that protein, that receptor doesn't have other binding site. Now, these binding sites are the amino acid chains generally. So you might have a protein that has 500 amino acid. All these 500 amino acid are potential binding sites. They are potential binding sites. And that is why if you are doing blind docking, you don't define your binding sites. You allow the binding to just proceed. Then it will begin to search for different binding positions. From there, you can get the one that has a better score. Yes. So, you cannot do a docking with a protein. Maybe I'm docking a receptor, and I want that receptor to bind at different sites, like to bind at different binding sites entirely. So the binding usually occurs at a specific region. It occurs at a specific region. What you may not do is that if you don't want to follow the protocol of concentrating on the active binding site defined by the native ligand, you can now begin to change the binding position, the binding site. So you keep changing the binding site by yourself and dock. Now from there, you may even notice that some of the binding sites that you altered might even give a better interaction than the proposed active binding site from the native ligand. So there is no one binding site for one receptor. There is no one binding site. Yes. In fact, most times what I do for the sake of my own work is that I subject my docking to different binding sites for my personal work. But for people who are interested in publication, they would like to see that you use a protein that has a co-crystal ligand, and they would like to know that this native ligand defines your active binding site. Some of the reviewers will not listen to your opinion that you don't want to work on this defined binding site. They want to use other binding site. They will tell you that the native ligand defines the binding site. That is for publication. But if I'm doing a personal work, I try to check other binding positions or other binding sites of my receptor. So there is no one binding site, and you cannot do these different binding sites at the same time. So it doesn't work at the same time. So if you want, you can set at this site and do your work, record your results, then you can come again for another redocking, set at another binding site. You can set them based on your grid bus dimension. We shall look at it based on your grid bus dimension. You can adjust the binding site and redock. So while you are adjusting the binding site, you are taking record of the grid bus dimensions, because it will be part of your report. In case of someone want to redo what you have done, then they have to focus on the binding site you have defined, on the SYZ value, the dimension you have defined for that binding site. So that is the only thing you can do if you want to look at traces of multi-binding sites in a receptor.
What else, sir?
All right, yeah.
Thank you very much. To make a kind of clarification concerning having a different binding site. If a protein is going to have different binding site, is it not that that protein is having multi subunits? so that each of those subunits are having their own active site. So that is why in docking we have to delete other subunits to retain a single subunit that is having one active site.
Okay. Yes. Now I made a case study of a protein that has 500 amino acids, 500 sequence length. Now, when you have 500 amino acids, you will notice that not all those, in protein preparation, we have to delete some chains. Now, but even when you leave one chain, maybe chain A or chain B, whatever chain you leave, you'll notice that that chain has multiple amino acids. But not all these amino acids in that chain that you left, we are involved in the docking. Now, when you generate your 2D, interaction or 3D, that is when you will now see the amino acids, the amino acid residues that we are involved in the interaction. You find out that most times, so you may even count like 10 amino acids in the interaction at that binding site, whereas the chain you are using has even up to 400 amino acids. So what happens to the rest? It means that the interaction just only took place at that region where you had the 2D amino acids. So there are other amino acids in that same chain you left that did not bother themselves in the interaction because you have already defined a binding site. So even when you are deleting some chains, you are not still, that does not still mean that you are defining the binding site. No, that doesn't still mean that there are still multiple binding sites in even that chain that you left. Because if you look at the 2D interaction, you don't expect to see all the amino acid in the chain you left in the 2D. You only see the one that took part in the interaction. So the rest did not contribute to it. They were just there looking at you. So we can also try redocking using other binding positions. So this is what I do in my personal work, in my personal study. Like if I want to develop a formulation, I try different binding sites before I can draw my conclusions. Okay, I saw Dr. Vincent, his hand was raised up before.
Yeah, thank you, Dr. Harry. Your response is apt, okay, and thank you for that explanation too, because the proteins is very large, and then of course you must define the area you want to target in that protein. So I think, to the second but last speaker, the only thing I wanted to add is look back to the original concept of drug protein interaction using Paul Ehrlich, okay, or the magic bullet of Paul Ehrlich. It's just like you look this way, fire a bullet, and you expect that the bullet will locate the target anywhere, and then of course interact with it. So yes, the protein is so large, but you have to concentrate on a particular area of the protein as your target. And then if you want to target other area, especially when you say, you know, uh, uh, enhancement of the activity of the protein by other, compounds there. So you can't just do it multiple, in one docking. Rather, the only best way to do is to move the dock. After you dock one, then you can go to another site of the protein, and then begin to dock. Even in your your blind docking, the blind docking at the end of the day must locate the target to which the interaction must take place. So basically, that is, but your answer was very beautiful. Thank you very much.
All right, I want to appreciate Dr. Vincent for that response. So it's good that we get these basics because it will guide us. Like we always say that the practical is not our problem. The problem is the knowledge behind what you are doing. So that you can boldly say, I know what I'm doing. All right. Ayofe, is your hand still up or the former one that was up?
Sorry, the former one.
Okay. So at this point, let us prepare our protein. Sorry, our ligand. So we'll come back. Okay, whether I should go back to ligand. Maybe let me just go straight to the protein since we already talked about the protein. Then we'll then come back to get those ligands we have screened. So because we are not going to look at network pharmacology at this point, which are leverage on literatures. So you know the topic you now want to work on, you know what you want to do. So but me, I'm using my own compound for high blood pressure. So I'm going to leverage on literature to see their own receptor. So we'll just look at molecular doctrine. Please, if your mic is on, you can mute your mic. Molecular doctrine of probably plants or molecular doctrine against high blood pressure. So we just search for against high blood pressure. So you just have to search and see if there are some work that has been done and antihypertensive. So I may not be doing that random search. I will just so this is molecular doctrine and antihypertensive activity of eupalintine 3-beta-D litopinoside isolated from whatever plant is that, Boheva diffusa. And if you look down, you'll be able to see here this abstract is very poor because they could not identify they use ACE. So but if we try to download this work, then we're able to see they use ACE, the angiotensin converting enzymes. So we can look at the study. Okay, let me come down to materials and method molecular doctrine. All right. So this is what they did. Now we can see that they are focused on the ACE. They are focused on the ACE and the protein they used. They just used one protein. That is a 1J37. Then they are looking at the inhibition of that protein. Then you can check for the control that they use. So but the control, you can now go online and search for high blood pressure drugs that inhibit the angiotensin converting enzyme. So that might be your focus. So maybe I will just focus on the ACE tonight. But in your own work, you can easily study as much as possible to get other pathways. Like for BP, you have the calcium blockers. We have the calcium blockers and others. So you can just get as much literature as possible. Okay, so get as much literature as possible. So for the sake of the training, I will just focus on one pathway, which is the ACE inhibitors. So I'll just focus on one pathway. So this is one way, like I said, you can get the protein. And the other way is when you come to this database, you just type protein data bank. You can see if you can type high blood pressure. You can, but I don't advise you working with this way. Coming to search your protein through this way. Or hypertension, you can quickly type hypertension. So I don't advise you searching through this way. It is, to me, it's not good. You can leverage on literature. You can leverage on literature. Okay. All right. So let's look at for let's just continue with that, with this code, with this code. Then I will give you the conditions to select a protein. Maybe you have gotten the protein you want to use. There are conditions if you are to go ahead with that protein. So we'll just open this protein. I'll just copy this code. So you can work with me as well, because I want to use my plan for hypertension. So I'll copy this code. I come back to the protein data bank. So I will see if this protein satisfies my conditions. I'll give you some of the conditions to select a protein. In fact, one of the conditions is that the protein must have a co-crystallized ligand. Must have a co-crystallized ligand. Okay, I want to see what they use in this study. Okay, this study also did the angiotensin converting enzyme, or morinda, and the protein they used, if you scroll down, okay, they use 1086. So in this study too, they used 1086, still on the ACE, 1086. I'm waiting for this to pop up. So we have two proteins now. We'll just check these two proteins, the one that will be appropriate for us to use. Why is network now disturbing me? I wanted it to come up with the search button around here. Okay, let's let me check my network. Just on this white. Okay. So I'll work with the two protein. I will duplicate. So I'll work with the other protein. So I will compare both protein. I can compare both protein to know which of them will give us the conditions that we need. Okay, so that is for this one. And this is for this one. Okay. So when they open, we shall look at them and see their suitability.
Please, what's the code for the
Let-
second protein?
The, you say
What's the code for the second protein from that
the
literature?
code is 1086.
1086. Okay, sir.
1086. I think the network is very bad this at this end. Well, oh, oh, this is how will I explain this thing now? When you are ready to use the internet, it will start disturbing you. Ah, this is a mess. Oh, yeah. This is a really, really big mess. Ah, I've checked the network. Just have to switch to. eh, what kind of nonsense is this? I'm coming. Let me see the possibility of maybe just switch to another system to see if it can be resolved. We are really so sorry for that. Oh, ah, okay. Thank God. Now let's look. Can we see the screen?
Yes, sir.
All right. I'm so sorry for that. Now, I'm going to see if I'm to use this protein now. This has already disqualified me because the protein that you have to use, you must check the organism that secretes that gene. So if the organism, like if the organism is not human, though not all will be human, but the AC enzyme is secreted by the human body. So that means the organism here should be human, Homo sapiens. So that is the first thing you look at. Now, but if you are working with a fungi or bacteria, then the organism here should indicate the bacteria or the fungi you are working on, since that protein is not secreted by the human body. So the organism here must agree with the organism that secretes that protein, that secretes that enzyme, that secretes that gene. ACE is produced in the body, so your organism must bear Homo sapiens. I don't care whether it is crystallized, but since it's not having Homo sapiens, I will just disqualify it. Yes, I'll just disqualify it. Then another thing you check if it is crystallized. So there's no indication if this 1J37 is co-crystallized. So it's not co-crystallized, though it has a captopre, but it would have been stated here most times if it's co-crystallized. But I will not work with this one. So let me check the other one. All right. So this 1086 already is co-crystallized and complex with lisinopril. So that means the organism is already co-crystallized and complex with lisinopril. So this lisinopril now is the native ligand that we are going to use. Then if you check the organism, which is the number 2 factor for you to choose a protein, is targeting the Homo sapiens. That is the target organism, so we can go with it. Then you check the resolution. Your resolution, I prefer between 1.5 to 2.5. In fact, the lower the resolution, the better the protein. So I can give you a range of between 1.5 to 2.5. So it's a bit okay, but the lower the resolution, the better the protein. So I can go with this 2.0, and that is point number 3. The third criteria: 1, the protein must be co-crystallized. 2, it must indicate the target organism. 3, the resolution should be much lower and not more than 2.5. And then the method of preparation must be S-ray diffraction. So it must be S-ray diffraction. So, and if you look at 1086, the 1086 protein was able to accommodate these factors. The only challenge you may have now is, well, thank God most of us are already into docking. This protein has just one chain A. So the preparation will not stress us. But I would have loved it if the protein has multiple chains, so that we can easily know how to choose a chain out of a multiple chain. But I'm going to just explain it. If the protein has multiple chains.
Hello, Dr. Harry.
Yeah.
The protein actually has multiple chains. If you go down, you'll see multiple. Yes, the others are chains apart from chain A. If you go down, if you scroll down, see
yes,
E
yes.
Chain C is chain E.
You mean the small molecules?
Okay. No. I think it's just a single chain. It's single chain.
Hello, sir.
Yes. No, go ahead. No, go ahead. Go ahead.
Okay.
I know that. Go ahead.
Okay. So if you now look at the macro molecule, we're just seeing chain A here, and it's the organism Homo sapiens. So for practice purpose, I would have loved using proteins that has multiple chains, so we can easily know the criteria to choose a chain. But let me just say it. If you have multiple chains, then look out for the chain where the co-crystal ligand is attached to. So you may have chain A and B. And if you look at the co-crystal ligand, we scroll down to small molecule, The cold crystal ligand is this man, the first one. This first one. So this is the cold crystal ligand, this. And if you look at it, is attached to chain A. This OT A means where it is being attached to. So it's attached to chain A. So assuming the chain has chain A and B, and here is just showing chain A, that means you are going to focus on that chain A that has the native ligand. Since the chain B does not have the native ligand, then you focus on chain A. Now, but there are cases also where your native ligand maybe is also attached to chain A and B. So what I do is, if the native ligand is attached to both chain A and B, then you look out for their sequence length. Their sequence length, I usually go for the longer sequence length. Then there are cases also, this chain A and B have the same sequence length, and the native ligand is also attached to both chain A and B. At that point, you can choose any chain of your choice because they all meet the same criteria. Both of them have the same sequence length, and the native ligand is also attached to both chains. So at that point, you can choose any one you want to work with. You can even try working with chain A before, then later working with chain B. Okay, so because they meet the same criteria, but you must check the chain where the native ligand is attached to. That will define which chain that you're going to use while you are working. So we are going to.
Sorry,
you can
before
I
we
have
leave
a
this,
question though.
before we leave this page, please. Now, what does this chain connote structurally? Is it talking about the subunits, or what does it really connote? I mean, the chain.
The what? Chain.
The chain, yeah. What does it connote? Is it the subunit
Okay.
of the protein, or what does it connote? I mean, structurally,
Okay.
chain, yeah.
Okay. It's just like some of us who put on chains. Now, when we talk about chain is a connection, amino acids that are connected. Now, if you look at the chain, like some of you might be putting on chain right now, you check that, you see that in the chain, it has different units connected together, different units. Now, those units are amino acids that are connected together that make up that protein. So now the chains now are different sets. So we have set A, set B, set C. And most times you even notice that some of these sets, which are this chain now, so the chain is connected amino acid. So we may have, now you're putting on two sets of chain just on one neck. You put on first chain, we are another chain. So we can say, okay, the first chain you're putting on in chain A, the other one in chain B. And in some cases, this chain A, these different chains, we may end up having the same amino acids. That is where I explained if they have the same sequence length. Now, in some cases too, these sets, these chains, which are connected amino acids, may be of different amino acids. They may contain different number of amino acids. The reason is just based on the mode of preparation, how they modeled the protein. They have to combine different chains together. But if you are doing homology modeling, where you build the protein by yourself, you don't have multiple chains. The only multiple chains you may have is the case of oligosaccharide, like some smaller micromolecules, some smaller chains, which are not purely the protein itself. But if you are doing homology modeling, you don't have multiple chains. So the ideology of these chains are just groups of amino acids that we are connected. That is what the chain is all about.
Okay, thank you. The other question is that on this page that is showing the properties of the of the protein, I can see under the detail column, you can see there mutation. Do we have in what's it called, receptors that have genetic variations in terms of
Okay,
variants?
yes.
Do we have to take that? Do we have to take that into
Yes,
consideration?
I skipped that. Please don't attempt using protein that says yes that has a mutation that can mutate. So we don't use proteins that mutates. We don't use proteins that mutate. So the mutation must be no. Thank you for taking me back to that condition. The mutation must be no. All right.
Sorry, my question is on that chain. So in case we have 3 and probably, I don't know if it's possible, and 2 equal, what do we do in that case? Are we going to pick the 2 chains that are equal and leave the other one out?
Like I said, you check. The first thing you would check is go down to your small molecules, then identify your native ligand. So when you identify the native ligand, check which of the chains where the native ligand is attached to. So that chain that has the native ligand is where you are going to is now where you are going to concentrate on. So if I have multiple chains A to E and my native ligand is in chain B, that means I'm going to delete other chains and only work with chain B. So, but if you notice that your native ligand is also attached to two chains. So if they are attached to 2 chains, you now check the sequence length of those 2 chains. If these 2 chains are of the same sequence length, you are free to choose any one because they have the same sequence length. That is amino acid the sequence. The same number of amino acid. But if you check these 2 proteins where my native ligand is attached to, you look at these 2 proteins and they don't have the same sequence length, you will know. One way you will know is that they usually split the chains. Let me see if this other one has multiple chain. So okay, like in this case now, look at this one. This one has chain A and B, and this chain A and B is of the same sequence length. Okay, so if the native ligand is attached to both chain A and B now, I can choose any one. But if I have another column, if I have another row, another ID, if you scroll down and you see another ID still under macromolecule, then they will show the chain, maybe C and D. So when you see 2 chains together, means that they have the same sequence length. But when the chains are different, so they have different sequence length. So if your native ligand is attached to 2 different proteins that of different sequence length, I usually go for the one with the higher sequence length. Yeah, I go for the one with the higher sequence length. So that is just a simple approach for you to choose your protein.
So for in this case now, like this one you just did now, that have the same, this chain A and B that have the same sequence length. So we're going to work with either A or B, or we're working with both.
Good, either A or B. Yes. That is if the native ligand is also found in A and B. But the one we are using tonight now just have only 1 chain.
Okay, sir.
Okay, so what you would do now is you can study the
Yes, sir.
literature. Yeah,
Good evening, sir.
yeah, good evening.
I just want clarification. You said if you have two chains and one is longer, that you go for the longer one.
Yeah.
So do you go for the longer one irrespective of the fact that it has a native ligand or not?
Okay, no, 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 no,
What if
no.
the longer one does not have a native ligand attached to it? Do you still go for it?
Okay, no, 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 no. Okay. I I said the first condition is go for the protein that has the native ligand. Yes. Go for the protein that has the native ligand. So in a case where I have two chains, and these two chains now contains the native ligand. So from your question, if one has the native ligand, okay, one is longer without native ligand, and the other is short with a native ligand, I will go for the one that has the native ligand. But when both of them all contains the native ligand, that will go for the longer one, because at that point both chains all contain the native ligand.
Yes, sir. Thank you, sir.
All right. Okay. I like the way we are getting clarified. Is very, very, very much okay. So you can take your time to look at the literature of this protein. The literature will tell you more about the protein, and your toxic-converting enzyme has a critical role in cardiovascular function by cleaving the carboxyl terminal. So this is part of the pathways, the pathogenesis of this protein, the mechanism of interaction. So you can do that for your own good. So what's the next step? The next step is to download the protein. So we come to download files. You click on download files, come down to legacy PDB format. So you click on legacy PDB format. It will download. So having downloaded the protein, the next stage you will do is, so this is the protein 1086. So the next thing you do is to prepare the protein. But then before you close this chapter of this protein, I would suggest you quickly download this native ligand, the synopri. So go and download the synopri before you close this page. So you can go to PubChem. You can go to PubChem and download the Listenopre. That's it from PubChem. So when you download it, you save it with Listenopre. Well, if you are docking, once you are working with PubChem, getting structures from PubChem, it usually downloads with their CID. So you are going to take note of the CID that has that correlates with Listenopre. But for those who attended our beginners course on computational chemistry, you can use the approach we did on Notepad. You can use that approach to now change the name of this native ligand, so that whenever you're using it for docking, you can easily know that you can change the name to native ligand or to Listenopre. So that when you are doing your docking, you just know that this is Listenopre. So you can change the name in Notepad. So that is the privilege for those who were able to join us in that training that we concluded last week. So why PubChem is opening for me to download my native ligand? Let me go to my download. So we are going to prepare this protein 1086. We'll prepare the protein. So next thing is your protein preparation. So I'm using Discovery Studio. You can use Pymol, you can use Chimeria. Well, I just feel okay with Discovery Studio. I have Pymol, I have a Chimeria. Maybe one of these days we'll just organize a training on how to play around those different visualization softwares. So at this point, I assume that we have our Discovery Studio already with us. So you can launch your Discovery Studio. You can launch your Discovery Studio because we are now going to use it for protein preparation. So we use Discovery Studio for protein preparation, for visualization, for to visualize your 2D and 3D, for superimposition. And I want to encourage us that don't do a docking without performing superimposition. It's very necessary for comparison and validation. So if you have launched your Discovery Studio, please just do well by letting me know. Have we done that?
Yes, sir.
Okay. So you can equally download Listener Pre also. You can also download it. So this is the CID code. You take note of that. You can equally download it your end because it will be part of our ligand. How wish we may even have the time to do docking. I'll be so grateful. But I don't think if we may. What is I hope this man is not speaking grammar. Okay. So if you have opened your I don't have strength for this thing. What kind of what's happening to NCBI? Is it today that I have class and network will start disturbing? Today that we have something serious to do. Okay, why that is going? Okay, thank God. So we can easily just download it, the 3D conformer, and so I will rename it. You just have to take note of this code. I would have showed us how to actually do the notepad, but I will leave that for the benefit of certain persons. So this is my native ligand. So I'll just call it my native ligand, maintaining this code 5362119 because I will use that to identify. So double click on your protein. Once double click, it will open in the Discovery Studio. So double click on the protein, it will open in this Discovery Studio. It's still loading. All right. So this is our protein. You can click this panel here, this drop down, this little arrow here to open up the panel of that protein. So what we want to do now is to prepare the protein. We want to do protein preparation. We have to change this black background to white to enable you to see what the protein has. So you can right click. When you right click, you go to color and you go to background. You click white. All right. So on this protein, we have these dots. Yes, this reddish dots you are seeing here. They are your water molecules. So there are 4 things you do here in protein preparation. There are 4 things you do in protein preparation. All these dots are the water molecules. So the 4 things you do is, number 1, you remove water molecule. Number 2, you remove heteroatoms. Then you delete some chain, and then you add polar hydrogens. So these are the 4 steps that we will take here in protein preparation. We are going to remove water molecules. We are going to, this is water molecules, the water. Then above it is the heteroatoms. It will be removed. So once you remove heteroatoms are atoms not part of the protein, like your small molecules, your native ligands, they will be removed because they are part of the heteroatoms. So when you click on water, you can click on water. There are 2 ways you can do that. You press delete in your keyboard, or you just look above, you see this S close sign, this close delete sign. Once you click on it, you notice that the water molecules are off. The water molecules are off. I can still see my lisinoprid here. So I'm going to click on heteroatoms and delete heteroatoms. So now, because I have just 1 chain, that is my chain A, I will not delete other chains. That would have been the 3rd thing that you would do. So finally, I will add polar hydrogen. And in adding the polar hydrogen, you go to chemistry. When you go to, you click on chemistry, then click on hydrogen. When you click on hydrogen, you will see add polar. Add polar. So you just add the polar, and it will be added. You may not see any visible change, but it will be added there. Although even if you did not add the polar hydrogen, if you are working with Pyrese does that automatically. If you notice that there is no polar hydrogen, Pyrese usually adds polar hydrogen before performing this docking. So at this point, I'm going to resave the protein. So we are going to save this prepared protein. I hope I'm not too fast, right?
No, sir.
Okay. So we want to save this prepared protein. So you go to file. You go to file. Okay, that means we may have hope of going to docking. Let's say that by Friday, we'll just conclude macrokinetics, and that will be the end of the training. So go to file, go to save as, go to save as, go to save as, then choose where you want to save the protein. Where you want to save the protein, mine will appear under my download. Then you change the file type these file formats, change it to protein data bank files. So change that to protein data bank files. Then to differentiate this prepared protein from the downloaded protein, you just have to actually put underscore hypertension. So that we know that this protein is for hypertension. Some persons might include PrEP, which means that it was prepared. But once I already include my hypertension, I know that I have prepared it already. That is for me. And this will enable me to search easily if I want to reuse the protein. So you save. So you have been able to save the protein, the hypertension, the one you have prepared, and that is what we shall be using to work with tonight. All right. So now that you are done with the protein, we are now going to go back to our ligand. So we have a native ligand now, so we can go online and search for our positive ligand. So you can easily search for FDA drugs that inhibits ACE for hypertension, because we are using the ACE, so we must look for drugs that also inhibit the ACE. So you can see FDA approved ACE inhibitors commonly used to treat hypertension include lisinopril, enalapril, ramipril, benazeprile. So which one do you want us to work on? So we'll just pick like two. We'll pick like two. So I will pick already. Lisinopril is part of our native ligand. So I will take the enalapril. I will take the ramipril. So these two, I will pick these two. Then I will download them. I will go back to PubChem to retrieve their structure. So I'm going to retrieve their structure. So for ramipril, I search. Okay, I know you are working with me. So I will download. I will retrieve the 3D structure of the ramipril. I can duplicate this and probably do same for the other compound. Why that one is green? I will now download my enalapril. So
Please,
this
sir,
is
how do you come up with that double download, that double copying? Sir, can you tell me about it? Because you copied two items at the same time.
my clipboard is already on. Like I already activated my clipboard, so I can copy multiple ones. Then with your Windows V will open up the clipboard, so you can click on the ones you have copied. So you just like copy this, copy. Then where you want to paste it, you use Windows V. Click Windows V. Windows V will now open up those things you have copied. Then you paste the one you want to paste. All right. Okay, so I will download pre 3D conformer. Then, okay, quickly, let me just change the name so that it will be easier for me to identify. Pre, right? Then my pre. So if I'm able to produce, if I have my own compound, at least I will call it pre. Since all of them are ending with pre. So let's wait for this to download, to show up. So we'll just download it. Why that is going, okay, that is great. We download. I'll just open up. So I'll rename. Sorry. I just did a mistake. This is for pre. Okay. So I've gotten my two positive like that, my native like that. Then I need my negative like that. So which disease do you think that really does not have much business with hypertension? Probably I can still work on that Hazima. So let me say FDA drugs. FDA drugs. Just pick one. FDA drugs for okay, FDA Hazima drugs. So I'll just pick one. So I'm using it as my native like that. So let me use donanemab. So I will use donanemab. So I will also download the structure.
Hello, sir. As you're done the social, I want to ask a question, sir. Can you hear me, sir?
Yeah, I can hear you.
You know, some of these drugs have a counter interaction. Some of them the side effects can lead to that disease you're talking about. Like for example, now some drugs now their side effects can increase like even though it's not meant for it, but it can increase the rate of high heartbeats, blood pressure. So how will you identify that? How will you, because that one now will also affect for me, I feel that it can affect the study you are going to do. So how will you, rationale that? Thank you, sir.
Okay, if I understand that me using this drug might counter my docking, right?
No, 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 not countering. Like for example, now some of these drugs that you want to pick may have some like counter indication. Like if you check their side effects, they say that it
Okay,
may increase hair. So how will you
okay,
identify? Because in that case, now it will affect the docking.
okay,
Like it may have positive negative result. I've forgotten what they call it in the research study anyway.
okay. I understand. Like the controls I'm using now, like the NLI and the ME, they might have side effects on the body. Yeah, but in
Sorry,
this
sorry,
case,
sorry. That's not the concern. The concern is that
the negative control that you are using,
Okay, yeah,
we're assuming that it should not have positive binding, positive docking properties with
yeah,
the receptor. That's our
yes,
assumption. Yeah, we want
yeah,
to
that's
choose a
assumption,
drug,
yes,
yeah, that should not have any binding property with the receptor. But again, what he is saying is that in case we choose a drug that is, though it is not used in hypertension, but
mm,
based on its side effects. So for example, we are using an Alzheimer treating drug. Maybe
yeah,
that Alzheimer treating drug, for example, is causing a side effect like cough. And the
yes,
cough side effect is brought about because it is inhibiting that ACE enzyme that we are
yes,
talking for.
yes,
I hope you understand now.
okay. Yes, in fact, this point you raised now. When I had a training with Genomat Institute, I was able to facilitate a training for them. And there was a question that was raised, and it's similar to this now. We are supposed to argue the use of negative ligand. Yeah, because if you look at it scientifically, it's not ideal. But why we choose to bring it up is that I've seen papers where reviewers will tell you, guy, you must use a negative ligand. And if you try to argue with them, you're on your own. But if you look at it scientifically, there is no drug that is meant for one disease. In fact, the idea is we want to see if, since this Alzheimer drug does not target the ACE, so we want to compare off-target interaction. But at the same time, don't you think, like what you just said now, don't you think that this drug we are using for Alzheimer can equally even be used for the treatment of hypertension? So let me even come above what you're asking, because your question is it may also target that ACE enzyme. It might target it for good. Maybe targeting the ACE enzyme might lead to a side effect. Yes. Even it might also turn out to be a better drug for hypertension. That is what we call drug repurposing or drug repository. So that a drug is used to cure malaria, that doesn't mean that that drug cannot cure other disease. There are a lot of publications on this. And one of them was seen during the COVID. During the COVID, people were testing chloroquine. Chloroquine is an anti-malaria drug, but we heard that it was effective during the COVID period. So they had to repurpose, redefine the drug. So it is possible, and we cannot tell from the docking. Yes, it is possible. You may even, because we are our focus is we are expecting to have a low interaction, a lower binding score from the native ligand. That is what we have in mind. So that we say, oh, since this is a native ligand, so and it's for Alzheimer's disease, it does not have a business with ACE, and we will not compare with our own ligand. If our own ligand also gives a lower binding score, postmate to the native to the Alzheimer's drug, they will not conclude that because our own ligand is giving us binding score similar to that of the native that of the Alzheimer's drug, so it may not effectively target ACE. That is mere assumption. A drug, we don't have one drug, one target. So a drug, I said it before here, that a drug will definitely have multi-target interaction. So even as we are trying to use it for as a negative ligand, we may end up seeing that it may even uh, uh, bind well with the ACE. Now, the binding affinity with the ACE now comes in two ways. That is what we're asking now. That binding might be for good or for bad. Yes, because interacting with the ACE might not lead to a side effect, or is high interaction with the ACE might be for inhibition. So it's a two-way thing. It's a two-way thing. And one way that you can easily know is if you want to now know if the high binding score on ACE, if it's for good, that is as an inhibitor on that enzyme, or if it will lead to side effects. Good. Now you have to now study effects generated. You have to study on that ACE. Does the inhibition of ACE leads to a side effect? So if you have done finding and you have seen that if a drug is inhibiting ACE, if it will cause a side effect, why? In fact, most times it's not even if you are inhibiting. For my last class, I heard with some students, if you have two proteins, if I have two targets, maybe I have target A, and I am activating target A. Now I treated target A as independent receptor. So I activated target A and it was good. Then I also have target B, and I also activated target B and it was good. Now, if I bring these two together, most times activating one target might even lead to the inhibition of another target, and that target was not supposed to be inhibited, but just because I have activated this target A, and that activation also led to the inhibition of another enzyme that was not supposed to be inhibited, that will lead to a side effect. And that is where the question is coming from. So you cannot basically tell all this just from this native ligand. You have to now study the connection between these two proteins. These two proteins, you study the connection. That is the essence of network pharmacology, because network pharmacology will even help you to identify off-target interaction. There is the last training I did December with some professors on MP. We noticed two enzymes, and from the third pathway, we noticed that these two enzymes are good, though they work together. But all our idea was activating the two enzymes. We now notice that activating these two enzymes will make the other one to produce a side effect. So that is to tell you that if we continue with this interaction, we just identified a side effect. But we are not going to take because of the side effects and reject the work. We have to note it down as side effect, because there must be side effect most times in your drug. So in conclusion to the question, this Hazima drug we are using definitely may end up creating a side effect by activating or inhibiting the ACE. And even the activation or the inhibition might also not even have business with the side effect. It might even end up becoming a better inhibitor. So for you to now differentiate between these two, you have to study these two targets, the target for the Alzheimer's disease and the target for ACE. If activating as the Alzheimer's target, if it has effect on ACE, that is the only way you cannot conclude that the binding score of your native ligand is actually inhibiting, is actually causing a side effect or not. So I've really said a lot, but that is the only way you can use to do that. So maybe network technology will be grateful for us. Yeah, because it will expose us to too many things. Donanap does not have a structure, so let me leave that and go for another one. There was no CID for it. Okay. From look of things now, we will just download and there is no CID.
Hello, sir.
There's no CID. Why?
Hello, sir.
Yes.
You can use some other drugs like
Let me just use. Let me use Donzipri. Let me just use this.
Exactly.
Yes. That one is a very common enzymatic drug. I think I've used that before when I was working on neurological disorder. Okay. So let's just see how we can succeed in. We have barely 4 minutes, but we have not downloaded. We have not settled our ligands. The ligand we screened. We still have to download the structures of those ligand we have screened. Or there's no need to even download the structure. We already have their structure. We will just have to identify their PubChem CID and work with that CID. So I download this. All right. So I will just have to save it with the name. Sorry. I have to, okay. So we have really succeeded in doing something tonight. I will call it donepezil negative control. So I will know that it's for my negative control. Okay. So we have settled everything about controls and proteins. So I think we shall fall back to our screen result now. So we shall fall back to our screen results. All right. Can you see my screen? Can you see the Excel file, the Excel sheet?
Yes, I can.
Okay, thank you. Here we, okay, first of all, let me cover up this because we are using 70% and above. So let me just indicate with a color. Let me just indicate this with a color. Okay. So what I did was we are after the ones with 70%. Now, There are two screening we did. We screened for toxicity and we said let's focus on we gave a benchmark of 70%, and from the 20 smart code we used, it appears only this one, only the first one. Let me zoom my screen. So it appears that only this first one, only the first one happens to have that 70%. But then the question will now be, doing my docking, will I dock based on the drug likeness properties, or I'm docking based on the results of the toxicity screening? Now, the toxicity screening was just to give you is to provide you with adequate information telling you that this is the percentage of abiotic compounds in this plant that may probably pose toxic effects. So maybe just now look at out of 10, out of 20, only just this one was able to pass our criteria. That means 19 out of 20 based on our criteria is will be toxic. Now, so it's giving you idea that that the percentage of the ones that are not toxic is very poor. Out of 20, only one was able to pass the 70% scale. Then the rest, 19, failed. So you should be careful now with this plant. Just for this 20 we did, you should be careful with this plant. But we will not dock with this only one result now. This is also to give us an idea of the potential toxicity of our plant. We shall concentrate on the drug likeness result, because we're not doing modification, we are not doing optimization. So if you decide to dock with the whole compound, maybe like as I said, some of you have 1,000 compounds. So if you want to dock with the whole 1,000 compound, you may end up docking with the ones that are not drug-like. And if those ones that are not drug-like gives you a good result, and you are celebrating with those ones that are not drug-like, and they gave you a good result, and you are celebrating, in reality, there is no effect, because they are not drug-like. They cannot be absorbed by the body. But your docking result shows that they can bind. That is simulation. But if you now focus on the ones that are drug-like, so any result you see there, it simply means that such results can be replicated in situ, because they are drug-like, they can be absorbed, they have the properties to be used as a drug. So that is the reason why if I'm doing a work on herbal formulation, I focus on the drug-like molecule. So for this one now, we should focus on these ones I colored purple. Yeah, the ones with purple coloration now. So we should focus on this. We should focus on this now. The next question that will come to your mind is, how will I get the structure? How will I now get the structures of this compound? Now, since there are few, you can go to PubChem using their smart code now to download them one by one. So you can download them one by one. So I'm doing this with my own data. So we can download them one by one with their smart code. Or since we already had, remember we had a file, we already have the folder that has all those structures. You can check for their CID code. You can check for their CID code. So from the CID code of this SMICE, you know, when we converted from structure to SMICE code, I think I have that file. It saved as it's saved with the SMICE code and their CID. So you can trace back with that file. If that won't get you confused, if you won't be confused with that, you can trace back. But to avoid confusion, I will just advise you to we download the one that passed. So we are going to download these ones. You can just copy the SMICE code, and it will not take time because you are no more downloading all of them. You are just picking the ones that are drawn like. So once you paste the SMICE code, you download the structure. So it won't take you that time. It won't take you that time. So because you are not downloading everything, but if you can trace back the SMICE code with their ID, then you can easily pick out their ID from those structures you have downloaded. Then use them for your docking. So at this point, we are going to, I think we have achieved little. So what is remaining now is docking. And then if there are 2D structures, then you can use your Open Babel to convert 2D structures to 3D. I think I will end with that, so that just give me more 5 minutes. Let me show you the conversion of 2D to 3D. So that when we now meet on Friday, what we'll just do is to do the docking and our post-docking analysis. So we'll see how we can use that. Yes, it's possible to use that 2 hours for Friday to do your docking and post-docking analysis, which is the pharmacokinetics. So let me see if I can have a 2D structure. I will just show you how to convert. So launch your Open Babel. Launch your Open Babel. So, okay, fine. Look at this structure now. Is in 2D. We are not going to work with 2D structure. We usually convert them to 3D. Although the conversion might take time, but we must do it. We must do it. You must convert your 2D to 3D. So kindly launch your Open Babel so that we can end with that. If you look at these structures I'm downloading, they are novel because there's no much information about them. The first one I downloaded 2017. The second one I downloaded 2019. I just pray that they will have a better result because they appear to be new and their content is still empty. All right, so let me just use these two I've done. The two I've done, let me use them to convert to 3D. Let me convert that to 3D. Okay. We have achieved a lot. Okay, this one too is still novel. the content is scanty, but I can see macroelectro and biochemistry, but was created 2017. It's still a bit okay. Not that much. Still 2D. Now, wow. Okay, so let me use these 3 molecules to show you how to convert. Now, let's open our Open Babel. Launch your Open Babel. Next 5 minutes, we should be running up. Please from 10:15. So, have you opened your Open Babel? Hello? Hello, can you hear me?
Yes, we can hear you.
Okay.
Loud and clear.
All right. Now, for us to use Open Babel to convert those 2D structures to 3D, make sure that your input format is at SDF. Sorry. And your output format too is at SDF. So if it was at SMI for Smart Code, you kindly change it to SDF. So import those your 2D structures. So import those 2D structures. You see that they are bearing a structure already. So we import them. Open. Then what you will do next is just don't click on convert yet. Under this convert panel, just scroll down, you will see generate 3D coordinates. Can you see it? Can we see it?
Yes, we can.
So check it, just click on it. Let it be checked. So once you have clicked on generate 3D coordinates, and your output must be at SDF, then click convert. So once you click on convert, it will take a little time because generating 3D coordinate takes time. It might take some minutes. In fact, I remember.
Hello, sir.
Yes.
Hello, sir. It didn't select the output folder. So I don't know whether it's an omission or it didn't select output folder.
Oh, sorry. Sorry. I didn't selected my output folder where it will save. Sorry for that. Sorry. Let me relaunch my Open Babel. Thank you. Where I was trying to beat up with time. So when we do this conversion, it might take little time. I think one prof at Benediction University, Okada, when we engaged her in that training, she complained that her 3D conversion has taken 2 hours. Just conversion has taken 2 hours because she loaded about 50 2Ds at a time. In fact, she got afraid and closed the Open Babel and started all over again. So I will just call it converted 3D. Let me call it converted 3D structures. Now, what this thing will do is that it will not convert them and keep them single. It will convert them and cluster them in one file. So all of them will be in that same file called the converted 3D structure. So they will just be there as one file. And that is another thing we shall learn. Clustering of ligands. So I check my generate 3D and then convert. All right. So it might take little time, but while that is going, we have questions, comments, and reactions. We can do that now while we conclude for tonight. So let me take your few questions again. Somebody just said that his own doesn't take much time. So glory be to God that your own has finished converting. Mine is still converting. So that should be different in our system. So whichever way, once it is done, we can keep the file for Friday class. Right. Any question? Yeah, we have question. We are quiet, so it means we assimilated the class very well, and it was maybe the class has a good absorption properties. That's interesting. Okay, so we are drawing closer. Yeah, we are drawing closer. So by Friday, we shall use first 1 hour for the docking, and while we now use the other 1 hour for pharmacokinetics. I have to take my time to explain each of the absorption variables, CACO2, PGP, all those things. We have to look at their implications, each of them. Then when we are done with absorption, we look at the distribution parameters, each of them, their implications. Then we look at the metabolism parameters, their implications, the excretion parameters, and their implications. That we will now end the pharmacokinetics. So far, again, I want to appreciate each and every one of you for finding out time to be around. Okay, let me quickly share my screen because mine has finished converting. So let me just show you.
While you are sharing your screen, the other time when we looked at the toxicity profile of the compounds, you promised to give us the tool to assess reproductive toxicity.
Oh, yes. Yes, we promised that. I need to do that before Friday. Even I also promise to give us other database for toxicity screening, so you can combine the data together. I remember I promised that. So these two, I will do that before Friday. Maybe tomorrow I will just release some of those data. Okay. All right. So can you see my screen?
Yes, very visible.
Yes,
All
we
right.
can see it.
So you can see what is saying. It's saying 3 molecules converted. If I go back to the folder where I choose, so it will show converted. So these converted 3D structures are those 3 molecules. If you attempt opening them with Discovery Studio, you will see the 3 molecules all in one. So these are the 3 molecules, 1, 2, 3. They were compressed, they are clustered into a single file. And that is one thing you do before we go for docking. Before we do docking, we are going to cluster all our ligands into a single file. If you're using Pirates, both the converted structures and the non-converted one, the native ligand will compress all into a single file. We'll call it clustering. We'll do that by Friday. Then we'll proceed for the docking. All right. Thank you, Professor Olabi, for that reminder. I really appreciate.
Always a pleasure.
We are starting a training tomorrow on Bioinformatics for life sciences. What we did here was just the part that concerns us. We shall be doing details. Our vision in that training is how to build a personalized database. Yes, how to build a personalized database. We have sent out mails for that training already. We will do that today. So tomorrow, for those who registered for that training, tomorrow we are meeting again for Bioinformatics for life sciences. I really want to appreciate each and every one of us here, especially the Uniben participants, courtesy of our professor. I want to appreciate everyone. So let's see how it goes. I believe we have the work we are doing already. We have a different disease that we want to work on. So we can do what we have done now with your own. So that is our idea. As we do here, you also replicate it with your own work that you are doing. So that at the end it will not be shoot up. In fact, it will even help you for your rehearsal, for practice. So thank you once more. I will say good night. This we have tried. This is 10:22. So thank you once again for coming.
Good night.
All right, good night.
Good night. Thank you for your lecture.
Thank you, Messi. Thank you.
Okay.
Good night, sir.
Thank you, my beloved friend. You are well pleased, Dr. Choba. Thank you so much. So let's get ready for next week webinar. It will be so interesting. The flyer will be out. All right, good night.